we'll have to. So, okay. For some reason, my Duo Mobile. Do you guys get annoyed with that two-factor authorization? It's still logging in, like it still doesn't work. I hate it. Whoever invented that should be punished. Uh, so anyway, let's move on with. I think we are most of us here. If we have time, Marisa will go over quiz. If we don't, and I think we will, we can do review next week. So last unit before midterm. So whatever we do next week, even I know midterm is not next week, midterm is on 15th, like week after, but whatever we do next week doesn't go on midterm. So this is the last one, okay? And we will have like a mini review, just I have to make your exam. Um, I almost thought that I have exam with Micro 315. I almost gave them your exam. That would be like a killer. Anyway, so we're still talking about microbial stress, and this one, in my opinion, is way easier than last lecture where we talk about stringent response and nutrient starvation, but there will still be molecular mechanisms that you will have to know. And again, I'm recording this lecture and it will be posted on e-class through you. Quick announcement about that. I forgot to upload one of the lectures and then I couldn't find it in my video, so you guys didn't have the lecture, then I posted later. But what you can do if that happens, if I forget or something like that, I will try not to. You can always go to my YouTube channel. Apparently, I have my YouTube channel. I didn't even know that something like that exists. I just convert my video to YouTube and I leave it over there, but it's public, so you can find everything over there. So in case I forget, it's easier if I post it and I will do my best, but if it's urgent, you can always go there. We talked about starvation in terms of stringent response and sporulation. Today we are talking about membrane stress response. And we will start with the temperature response. We will cover cold and hot temperature stress response. And we will end up with pH adaptations, which are really interesting. While salt response is very short, like only one slide, and it's not hard to understand. But temperature stress is, in my opinion, one of the most significant and most interesting, and from evolutionary perspective and from physiological perspective. Although pH is also, it's hard to decide. They're all great. So what happens in higher and lower temperatures? So as you know, microbial growth rate will increase with the temperature, but each microbe has their own specific temperature for growth. This annoys me a lot, so I will have to somehow move it like here. Growth rate gets increased after the optimal temperature is achieved, and in optimal temperature, growth rate is the highest. The growth will slow down, and after certain high, so after going above that optimal high temperature, growth rate will slow down, and cell cannot survive. Why cell cannot survive when temperature goes above optimal temperature, when it's higher? Because enzymes that control growth are proteins, and they tend to denature it at higher temperatures. On the other hand, when cell is at lower temperature, again, growth rate will slow down. So you have some optimal temperature, and after temperature is decreasing below the optimal temperature, the growth will slow down. And eventually the production of whole molecules will stop and cell will not survive. So maximal and minimal temperature of survival exists for each organism. That means that your organism won't die immediately if you increase temperature at 40 degrees and optimum is 37. But if you go up to 80, then yes, because proteins will denature. However, bacteria can develop certain adaptations that will help them to survive high and low temperature, and some of them actually evolved to live in extremely hot and extremely cold environments, and they're called temperature extreme. Again, just a graphical representation of what I was talking about. So you have some optimum temperature when enzymatic reactions are occurring at maximum possible rate. That's the orange box. And then when temperature is below that optimal temperature, reactions are slowing down. So you have minimum and maximum. So that optimum has some plus minus, it's like plus minus standard deviation. So beneath that minimum, membranes getting to be gelled, transport processes are slowing down until they completely stop. If you go to above that max temperature, proteins will denaturate and cell will not survive. But as I said, bacteria, actually microbes, can develop certain adaptations to survive cold and hot temperatures. And in terms of cold survival, in terms of both hot and cold, in terms of both temperature stresses, they are adjusting their phospholipid membrane to fit with existing environments. So what is happening in the membrane? So as you know, cell membrane has those phospholipids that are made of fatty acids and glycerol. So if you remember from Unit 3, we talked about fatty acids being saturated and unsaturated. When they are saturated, the orientation of fatty acid will be trans. When they're formed in that trans and oil coenzyme A, just one step before you get acyl coenzyme A, that is done by Fab-I enzyme, final reductase. That's Unit 3. So that's happening in normal conditions. But when temperature is lower, what's happening with cell is decrease in melting point of phospholipids, which means that they will, a cell membrane will remain fluid at lower temperatures. And lipids, butter and oil, are actually the best illustrator how same type of lipids can be different based on the structure of fatty acids. So if you have a butter, which is like a solid one, it has very high level of saturated fatty acids. So that's why it's solid. If you look at the oil, oil has a rich content of unsaturated fatty acids. That's why it's a liquid at room temperature. So what is different between these two lipids is the melting point. So at colder temperature, you will have decrease of melting point of cellular lipids. In terms of hot environment, as you can imagine, situation is slightly different. So again, palmitic acid is saturated, unsaturated is octadecanoic acid. I don't really ever ask students to know the name of these and these fatty acids. You just need to know that at cold temperature, there will be more unsaturated fatty acids, and on hot temperatures, high temperatures, will be more saturated fatty acids. It doesn't really mean what's the name of the fatty acid. So again, high level of saturated, lower level of unsaturated when cell is adapted to hot environment. So, and this saturation of fatty acid is occurring in the fatty acid biosynthesis. So if you remember, there is a different Fab-I enzyme that is going to operate differently when it needs to form trans versus cis bond. And that's what provides adaptation on different temperatures. So, but that is when we talk about phospholipids that are generated, like fatty acids that are generated. So when cell is adapted at a low temperature environment, it will keep generating more unsaturated fatty acid. But what's happening with phospholipids that are already in the membrane, like that already build the structure? So again, cell will have an enzyme that are expressed. Cells have enzymes that are expressed at different rates at different temperatures. So after temperature, when temperature drops, after temperature stress, these enzymes that will regulate level of saturation and saturation will integrate themselves in membrane and modify those already existing phospholipids just to make sure that cell will survive. Yes, there will be still more synthesis of unsaturated fatty acids if we talk about cold environment, but also there will be those enzymes that will make integration. So they will literally insert themselves into the cell membrane and modify those existing phospholipids. Under cold stress, higher level of unsaturated fatty acid, which increases membrane stability, rigidity. And one of the enzymes that is very important for this is the enzyme that is part of two-component regulatory system, which we covered in Unit 3. It's called the integral membrane protein. what this protein is doing, this is it's kinase. So it's DSK is doing autophosphorylation upon the cold stress, and that undergoes two component system regulation. So what that means, DSK gets autophosphorylated at his level, and then uh, when it's activated via phosphorylation, uh, that will cause phosphorylation of downstream regulator DES-R, and that DES-R, which is the response regulator, and gets phosphorylated on aspartate. So the one in membrane on histidine, the one in response is on aspartate. That phosphorylated DES-R will increase transcription of genes for desaturase, the enzyme that will introduce saturation into the existing phospholipids. So again, once when desaturase is transcribed, it's controlled via DESKR two-component system, desaturase will modify existing lipids. So once when there is enough unsaturated phospholipids, fatty acids to survive cold stress, this KR system will get dephosphorylated via phosphatase and slow down the system. So the one that is sensing changes in environment will be that K that is in membrane. So that K is sensor that actually modifies existing phospholipids. So again, this is for existing phospholipids, not for newly synthesized. In archaea, it's slightly different. In archaea, as you know, they're really extremely adaptable, and they survive. Many extremophiles belong to the archaea, and this class of archaea, Crenarchaeota, is famous by knowing. So it has that cyclohexane ring in lipids, and that increase in so more these cyclohexane rings, their increase is suggesting that it's a higher temperature. So in archaea, they mostly have adaptation on higher temperatures. So what people who are doing ecology often measure is this TEX86 index, because crenarchaeol, lipid present in archaea, has 86 carbons, so it's a really big molecule. So higher index corresponds to the higher temperature. So this TEX index is a structure that can be used to determine how much temperature increased in the ocean in past years. So easy peasy, right? Okay. Now let's go to the acidophile and alkalophile response. So acidophilic response, first thing that you have to distinguish here is the differences between real acidophile and acid tolerant microbes. So when something is acidophile, that means it evolves to live at low pH, which means if you put it on neutral pH, it won't survive. When something is acid tolerant, that means that it learned to live, like it developed certain adaptations to live at low pH, but once when you put it in neutral, it will be able to survive because it will just not increase transcription of acid tolerant genes. So this is the big difference that students very often have a hard time understanding. So if something is acidophilic, it has very specific features. If something is acid tolerant, it has specific adaptations. So acidophile will survive always under the extremely low pH, and it's usually very extreme. So what is common for those two types of microbes is that both of them modify their cellular membranes to be able to tolerate high level of protons in environment. Just some of them evolved that long time ago, and some of them will learn how to do it and develop certain adaptations. So all modifications for acid or alkali, so the same is with alkaliphiles. So acidophiles live at low pH. Alkaliphiles live at high pH, but both of them, what is common for both of them is that they all develop adaptations that are on the membrane. So specifically membrane related, and some of them are related to the like intracellular cytoplasmic comp. But it's mostly membrane related. The whole point of developing these adaptations or features is that cell must learn how to maintain its own neutral pH level, which means to protect from high input of H plus or H plus ion, depending on in which environment it lives. So why would that be? Because so the reason why cell needs to maintain cytoplasmic neutral environment is because the optimum pH at which all molecules work is neutral. So what cells are doing if they are living at low or high pH, they are developing certain membrane adaptations to prevent certain entrance and exits of molecules from outside and inside so they can maintain internal pH. So the point is that they're trying to maintain pH homeostasis. And what they are doing, they have proton pumps, of course. What is proton? Proton is acidifying molecules. So more protons means lower pH, high acidity. So acidophiles, so those organisms that live at low pH, they have certain proton pumps. Those are integral membrane proteins that export H plus ions from cytoplasm in environment. Why is that? Because if they live in low pH environment, means that's an area that has a lot of protons. So when those protons tend to enter their cytosol, they tend to export them through those proton pumps so they can maintain internal pH environment. And this is just what I just said. So basically, they are putting an effort to get rid of extra proton ions that came from the outside because they're trying to maintain their cytoplasm. On the other hand, those microbes that live in very high pH environment, they're called alkaliphiles. That means that they live in the area that has a lot of OH ions. So they still have proton pumps, but what those proton pumps do, they import hydrogen ions from the environment into the cytoplasm just to neutralize incoming OH ions from external environment. So all point is to maintain neutral cytoplasmic level. Whether they will develop proton pumps that will export protons that came from the outside, or which is for acidophiles, or they will develop proton pumps that will proton importers actually that will import protons from outside just to neutralize incoming hydrogen ion. It is all about maintaining pH homeostasis in cell. Acidophiles are the only group of organisms that have something we call flipped membrane potential. So what means flipped membrane potential? If you remember when we talked about proton motive force generation, we mentioned that in cell, you always have that proton gradients where there will be more positive outside and more negative or less positive inside. And that's what makes membrane potential or delta psi. In case of acidophiles, that delta pH actually is flipped, which means they have more positive inside and more negative outside. So that is their way how they maintain internal cytoplasmic pH. And these guys don't actually rely on delta psi, so they don't rely on electrical component or proton motive force. They rely on delta pH, on chemical component of proton motive force. Alkaliphiles, on the other hand, they have exactly same membrane potential. They don't have that flipped potential, but alkaliphiles rely on delta psi, which is the electrical component of proton motive force. They cannot rely on chemical component. Unlike neutrophiles, organisms that love neutral pH, even though they can learn and adapt how to live in lower pH, neutrophiles would rely on both delta psi and delta pH to energize their membranes. So how neutrophiles maintain homeostasis? Again, now we are talking about neutrophile organism that evolved and likes to live in normal, like pH 7, 6.8 pH, but now let's say you want to adapt that organism. You want that organism to become acid tolerant, like Marissa is doing, like Rachel, one of my students, is doing. So you're literally teaching that organism to become acidophile, but it is not true acidophile. So it's still going to rely on delta psi and delta pH because it is neutrophile. It won't have flipped membrane potential. So what will be some of adaptations in neutrophiles that will develop if your neutrophile learn how to be acidophile? So depending whether you are, we are talking about microbe that respires, which means using oxygen, or microbe that is, or terminal electron acceptor, or we are talking about fermenting microbes that are relying on substrate level phosphorylation. So, and we will cover both of these types of respiration. It will be more clear when we talk about metabolism in next week. So adaptations in neutrophiles that are developing in respiring microbes will be a very significant amount of produced cyclopropane fatty acids. What that means is that, for example, now when Marissa is adapting her strain, if she wants to make sure that her strain is really adapted, she can do analysis of fatty acids through fatty acid methyl esters, and she will likely be able to see a lot of cyclopropane fatty acid because that organism eventually develops cyclopropane fatty acid to protect itself from high level of H plus ion. Another gene that will probably be expressed will be hydrogenase. Why hydrogenase? Because it will move protons outside. It will simply pump it out, so to prevent entrance in the cell. So hydrogenase usually lives in periplasm. That's where the first barrier will be. Commonly, those neutrophiles will develop carboxylases, and they will couple that carboxylases with antiporter. What is that? How is this functioning? So again, remember, they're trying to get rid of the protons that are coming from the environment. So they will have antiporter that will kick out the proton from the inside, but it will be literally antiport. So proton transporter will export hydrogen and import glutamate. Why glutamate? Because glutamate will be decarboxylated with this carboxylate. So literally, antiporter and carboxylate are working together. So antiporter export proton, import glutamate, carboxylate comes decarboxylated, and prevents the cell from acidification. What is happening with fermenting microbes? Fermenting microbes have a little different structures, so they increase level of fermentation that consumes protons. So they're trying to make more consumption of malate to lactate, so they produce more lactate just because they want to consume more and more protons, so they can prevent protons from coming in. They have this ATP, specific type of ATP synthesis that will consume protons and bring them back to cytoplasm. Simple preventing like acidification, but still maintaining operation of metabolites. So this was for acidophiles. What will happen with alkaliphiles? They're much easier, but they have interesting features as well. So they will develop something called sodium proton antiporter. So think about this. Alkaliphiles will be exposed to the high level of OH ions, and OH ions will tend to come into the cell. So they had to make sure that they bring in something that will neutralize potentially high pH because of a lot of hydrogen ions, which means that they will develop this antiporter that will kick sodium out and switch it for protons. So it's like antiporter. Sodium comes out, proton comes in. And proton pumping will be down-regulated because they don't want to lose protons from cytoplasm. Alkaliphiles will not rely on protons for proton multiple generation, funny enough, because they still called PMF. They rely on another single positive ion, which is sodium, Na. And they have another antiporter, which will deal with potassium. And again, potassium will be exported and hydrogen will be imported to main cytoplasmic pH. But so these were all neutrophilic adaptations. So these guys, so Marissa's babies will have all these adaptations once when she gets it adapted. And it's a beautiful strain. It is. It is. Don't cry. It's nice and pink. It's a high maintenance, like a Barbie, right? It's a nice pink, and you have to buy her cabrio, or she won't adapt to low pH. Yeah, come on, it is. It's really beautiful. You should come to see it. It's all pinky. Everything is pink. Everything. Um, Really nice pink, and it won't break. Whatever you do. Uh, but this was all over acidophile. Like neutrophils that learn how to be acidophile. But what if something is really acidophile? Something that learned to be like evolved, not learned. I should not say learned. These guys learned. Something evolved to be acidophile. If that's the case, then they have membrane fully impermeable for proton, and their PMF will rely only on delta pH. Again, and remember, the big difference between a neutrophile that develops adaptations, the one that Marissa and Rachel have, they will have proton multipolars relying on delta pH and delta psi and normal membrane potential. Real guys, those that really are acidophiles, like a real thing, they will only rely on delta pH because their membrane potential is flipped. They have that very famous Donan potential that I will talk about in a few minutes. But their membrane is fully proton impermeable, and in order to make membrane proton impermeable, what they do, so think about this, you want to prevent something positive to come into the cell. So you want to make your membrane impermeable. What will you do? You will accumulate tons of positive charges, like just right on the edge of the membrane, because positive, they can't come in. So they will protect from the proton entry. And this is why cell is importing potassium via that KDP transport. You remember we talked about KDP EA transporter, like when it's importing potassium, getting phosphorylated, it has two components. So cell, that KDP transporter that we talked about in unit 3 is now going to be very phosphorylated to import potassium because it has to maintain that internal positive charge that is called Donan potential. That is why we see flipped membrane potential in real acidophiles. Like it will be more positive inside than outside. This is very important. Donan potential actually refers to that flipped delta psi, more positive internal, more negative external. And that's done through the potassium input usually. Again, be very careful about that. Potassium, if I ask you about increased activity of potassium channels, is that referred to the real acidophile? Be very careful what is the next in the question. Because it depends in which direction potassium goes. Potassium also can go out. So you have to be very careful about that. I know that was always confusing in the exam. So again, there will be no PMF. So it's not it won't be PMF, but PMF is relies on different components. So cell is respired and pushing protons out. So they will accumulate potassium inside of the cell. So this positive charge will make up for that protons pumped out in order to prevent cells from acidifying. So that resulting delta psi, basically there will be zero electrical component. The cell will import potassium ions and export hydrogen ion. So that's why you cannot rely on delta psi. You cannot rely on electrical component. You have to rely only on delta pH. Very often, in order to establish Donan potential, cells are producing certain polypeptide that will maintain pH in cytosol, again, just to prevent acidification. So what are mechanisms? Again, protein production, peptide production can be one of them used to maintain Donan potential. So what are cells doing there? They are doing potassium transport via that potassium KBP channel that we talked about. They make membrane filings and number of protons, and they maintain their pH through active export of protons. So they will have very active export of hydrogen ions, and this is this is common between neutrophile that learn how to be acidophile. They will all have proton exporters. And in cytoplasm, there will be enzymes and the proteins that will bind, like sequester those hydrogen ions that come in, just to prevent acidification of membrane. And yeah, they can also have multiple DNA repair mechanisms that because DNA gets damaged on the low pH, so they have repair mechanisms to fix macromolecules. So you remember how I talked about that decarboxylase that can be developed as as a response. It also exists in real acidophiles. So what the reason why that decarboxylase is developed is because it consumes the decarboxylase consumes protons to produce CO2 and H2. So remember when we talked about it a few slides ago, that is a proton antiporter and decarboxylase in neutrophile that learns how to be acidophile. So that can also be a mechanism that real acidophiles use to maintain dominant potential. And phosphate buffering in so some phosphate buffering in cytoplasm is used just to consume protons to remain pH stable. So basically the point is to get the molecules in that will reduce the amount of protons and stabilize pH. And one of the most interesting physiological mechanisms in terms of acidophiles is Helicobacter pylori. Did anyone ever have it? Thank God. It's the worst possible balance that you can have in your gut. And I will tell you how it actually. So Helicobacter pylori is not acidophile. It is neutrophile, but it is the example of smart neutrophile, which means that Helicobacter can actually survive in your gut even though the pH of your gut is actually very low. So normal neutrophile would not be able to survive, but Helicobacter is everything but normal. So what Helicobacter does, it actually senses when H+, so when you get your Helicobacter in your gut, like your stomach is very acidic, very low pH. So Helicobacter feels that low pH, usually below 4, in their periplasm. The way how it feels it is through sensory kinase in membrane. So this is so the way how we say that the way how Helicobacter feels low pH of the gut is through two component regulatory system, which again has that autophosphorylation and the dephosphorylation. So and then that causes phosphorylation of response regulator in cytoplasm. And that phosphorylated response regulator, again, after Helicobacter sense low pH, will go all the way downstream to upregulate. So that phosphorylated response regulator will increase transcription of genes that will code for the enzyme urease. So what will this urease do? It will urease that now Helicobacter makes, triggered by low pH, will now degrade urea that exists in human gut and it exists when we digest food. And urea will be degraded to ammonia and carbon dioxide. So literally Helicobacter will develop enzyme that will help them to buffer their own system. So when they develop that urease and urease degrade urea to ammonia and carbon dioxide, these buffering compounds will consume protons from our gut and it will happen in cytoplasm, which means Helicobacter will now maintain its own neutral pH and it will be able to survive. So again, Helicobacter is not acidophile, but it learned how to live in our gut through transcription of urease and buffering their own components. Any questions? Well, I even think I will finish this before so Marisa can go over quiz. What about alkalophilic response? This one is easy. This one is really easy. So if you remember, we talked about acidophiles, how they have flipped membrane potential and they rely exclusively on delta pH. Alkaliphiles do something different. So again, alkalophilic organisms live at very high pH, and at very high pH, we will have a lot of hydrogen OH ions, not hydrogen, OH ions in cytoplasm. So those organisms live optimally at pH like 10, 11. That's very high. Like neutral is 7. This is like extremely high. And the point is the same like in acidophiles. The whole point is to maintain cytoplasmic pH through different membrane and cytoplasmic modifications and to maintain biochemical stability of molecules. So the risk that these guys, alkalophilic organisms, face with is to lose protons in cytoplasm. So if they export protons actively like a normal neutrophilic organism would do, OH ions from the environment would come in and they would alkalophile their cytoplasm so they don't have neutral environment. And they're trying to prevent that from happening. So the chemical component of PMF, which is delta pH, will become more negative as the external pH increases. So that means that they cannot rely on chemical component, delta pH. So acidophilics actually rely exclusively on delta pH and they maintain donor potential. These guys actually rely only on delta psi, and which is electrical component of proton motive force, and in order to energize their membrane. So remember, they should not pump out protons. So if they energize their membrane through generation of proton motive force when they export hydrogen, that will be very high risk for their internal cytoplasm. That's why they have sodium motive force. So their pumps rely on sodium motive force to energize the membrane. And it's a very specific adaptation that exists only in neutrophiles, in alkaliphiles. So again, they still use electron transport chain to create energy, but they need to conserve protons that would be generally released. So they pump out sodium ions to generate proton or sodium motive force. One exception is ATP synthase. That enzyme in electron transport chain actually relies on protons, but all other transporters rely exclusively on sodium. So only ATP synthase. So the most important adaptation in alkaliphiles is antiporter, sodium hydrogen antiporter. And you can imagine logically, it is importing proton and exporting sodium outside of the cell. As I said, very easy. The example of alkalophilic organism is Bacillus. Pseudophymus something, don't memorize, know that it's only specific Bacillus. So this one is creating, this one has alkalotolerant machinery, so genes are actually regulated based on alkalinity of the system. So this organism will have a high level of sodium proton antiporters. And another special adaptation that Bacillus has, alkalophilic Bacillus has, is a specific quinone type. So, and if you remember when we talked about electron transport chain, we talked about quinone in complex 2. So these guys don't have quinone, they have menaquinone, and they use menaquinone to establish proton motive force. ATP synthase, again, this one is the only one that relies on proton motive force. It captures protons at one. What it does, it captures protons at one time and holds them in the membrane until there is enough of them to establish the gradient needed to phosphorylate ADP to ATP. So it still prevents us. It still kind of saves those protons. It captures them one at a time and holds them until it's enough. So it's still one specific adaptation. So it does use proton motive force, but collects them like one at a time. But mostly proton motive force is, well, it shouldn't be said proton motive force. The motive force to energize transporters is based on sodium. ATP synthesis is the only one. But the most important to know is that sodium hydrogen antiporter is involved, but still all transporters use sodium multiports except ATP synthase in the electron transport chain. And there is a menaquinone, which is a specific adaptation in alkalophilic organisms. Just briefly, comparison between acidophiles and alkaliphiles. They are common in some areas, like they both rely on specific membrane and cytoplasmic components, but they rely on different components of protein multiphores. So acidophiles will rely on delta pH, not delta psi, and the membrane, they will maintain donor potential through importing potassium. They have a non-permeable membrane for protons, and they develop compounds inside of cell to consume protons, like decarboxylase. Alkalophile rely on electrical components on proton multiphores, which is delta psi. Sodium is used to energize transporter, except for ATP synthase, which one is using protons. And the main adaptation is sodium hydrogen antiporter. And I would add here menaquinone, which is a different version of quinone in electron transport chain. And the last but not least is salt stress in microbes. So this is where people usually get confused between potassium channels. So make sure to understand when is potassium involved in salt response and when it's involved in acid stress. One of the common adaptations in, so what means salt tolerant microbe? Microbe that lives with a high level of salts, like something that has a lot of osmotic compounds dissolved, like a lot of sodium chloride, for example. So, and you can see that in E. coli. When you expose E. coli to the extremely high level of sodium chloride, E. coli will rapidly produce molecule called ubiquinone 8. So don't be confused. This is not menaquinone. This is ubiquinone 8. And ubiquinone 8 will integrate itself into the membrane to increase thickness and hydrophobicity. So this would be one of the adaptations of salt stress that you would see if you are adapting bacteria at high salt stress. What ubiquinone 8 is doing is a specific interaction with the phospholipid chain, and it's promoting the linking between bilayers and making a membrane like a glue, like sticky like a glue. And the whole point is to make that so sticky and preventing from sodium chloride to come and poison the cytoplasm. Again, same principle. They're trying to maintain internal homeostasis. Another adaptation, very famous in salt response, are aquaporins. The name tells itself. Aquaporins are transporters that are allowing passive influx of water to regulate level of water based on stress. So simple to dilute levels of salts that tend to enter from the environment. So one of the other common responses to salt stress is increased amount of those aquaporins because they get transcribed, they integrate themselves in the membrane, and then they allow water to come in, because if some salt came out from the external environment, more water influx will dilute it and enable cell to survive. Except of ubiquinone 8 and aquaporins, another high salt adaptations in microbes, and this will be something, so why glutamate catabolism? So it will be way more clear when we talk about nitrogen metabolism in Unit 7, but one of the salt adaptations is to reduce glutamate metabolism. So why? Because glutamate is amino acid. Glutamate catabolism is connected tightly to potassium. So this is where confusion can happen. So when there is osmotic stress, like high salt, cell will accumulate potassium just because that's a part of the salt stress. So glutamate is needed to balance high potassium level, right? Again, now this potassium comes from the stress. So it's salt overloaded, and potassium salts can be a part of that. So in that moment, cell will need to use that glutamate that it's made. That's why it has to suppress metabolism of glutamate for other purposes. So it has to use that glutamate inside of the cell to just create potassium glutamate that will that will have protective role, stabilize DNA and protein. So it's not like reduction of glutamate metabolism. It's not like glutamate won't be produced. No, actually, glutamate is quite important. Glutamate is there to form glutamate potassium, but glutamate in that moment has to be used only to make a complex with externally derived potassium. Another adaptations are accumulation of intracellular compounds like trehalose. Why? Because simple thing about diffusion. So if you have very high level of salt outside, that means you have lower salt level inside of the cell. By the rule of diffusion, solids will move from higher to lower concentration until concentrations get equal. So if cell increases concentration of some other solid like trehalose or something else, that will prevent outside salt input. It's like a barrier. So not everything is logical, like from the purpose, like if you think from physics perspective. And another thing is they can increase water influx through aquaporins. So if salts accumulate, they will increase aquaporins, then water will come in and dilute salt, so the concentration is lower, so internal homeostasis is maintained. Oops. And another adaptations are some specific enzymes that can mediate how much solids are present in between in and outside of the cell. So again, it's like a buffering compound in cytoplasm. So in summary, just briefly, both bacteria and archaea respond by membrane changing of membrane rigidity into the temperature changes. So either it's saturation, desaturation, or in case of archaea, you have that TEX index. So more of that TEX protein, TEX molecule, not protein, sorry, TEX molecule will mean higher temperature. Bacteria respond to the pH stress by either keeping protons outside or keeping protons in. So if they're keeping protons out, that's acidophilic response. If they're keeping protons in, it's alkalophilic. Bacteria respond to salt by changing the thickness of their membrane, or they're generating certain salts inside of the cell, or they're developing integral membrane transporters to dilute internal environment. And all sensing is accomplished at the membrane. That's pretty much common for everyone. And that would be all from me for this class.